Welcome to our first lecture in the cell division chapter. This chapter is going to basically be talking about the life cycle of the cell before we get into anything about cell division. So let's get started. Um, okay, so a couple of, and I don't know if this is translating well to the recording because I think that there's some lag, but these animations just show a couple of examples of cell divisions. This is one cell dividing into two. Here's the nucleus dividing, and then the cell will divide afterwards. See, there's the cell splitting. Um, so it looks like this is just mostly something that we would call cytokinesis. All right, in this chapter, there are three main topics. The first one is the life cycle of the cell. That will be followed by how the cell divides by a process called mitosis. And then lastly, we'll look at how a cell divides by the process of meiosis. They're very similar, but there are some crucial differences between them that really depend on the type of cell that's dividing and what the purpose of that division is going to be. All right, so before we get into that, let's talk about types of cells. There are basically two broad categories of cells. One is your body cells, things that make up cells of your body, your skin, your liver, your brain, your heart, your muscle tissue, all that kind of stuff. Those cells are called somatic cells, and they divide by the process of mitosis. What this means is that those cells, when they divide, they're going to be genetically identical, unless, of course, there's a mutation. But for the most part, if the process is done correctly, they're going to be genetically identical. Germ cells, on the other hand, make up a very small percentage of of your body cells. They're really only your sex cells. They're going to divide, and they'll produce what are called gametes. If you're a male, this gamete is called a sperm, and if you're a female, the gamete is called an egg. Germ cells divide by the process of meiosis. It's slightly different, and the end result is that each gamete, each cell that's produced after division by meiosis, is going to be genetically unique. And that's going to have ramifications for a population and its offspring. Just a couple of interesting notes about cells. Different cells last different amounts of time. Skin cells typically replicate 65 times a day. They go through division very, very quickly. Conversely, pancreatic cells take a very long time to divide, about 1 year, so they have a long lifespan. Red blood cells can divide in 4 months, and after a certain number of divisions, a cell will know to self-destruct. This is how your body doesn't become just one huge mass of exponential growth. So after, we'll say, 3 or 4 divisions, there's something in the DNA code that will tell your cell to self-destruct and reuse its parts for a different cell. Interestingly, it can be said that 99% of the cells in your body were not intact, were not an actual part, were not a cell 9 years ago. So at the cellular level, we're all pretty young. One neat thing about lobsters, why this lobster here, is because lobsters have an interesting, when our cells divide, there are things that kind of cause degradation, and that's what results in aging. Um, but lobsters don't do that. They exhibit this thing called negligible senescence, which means that their cells, when they divide perfectly, ideally, and there's no degradation, there's no loss of function, so they never really age from old age. They don't get tired, they don't get, their elasticity and their muscle stays tight. Um, so they never really die of old age. The only way a lobster will die is of disease or injury or being captured and eaten. But lobster cells are pretty interesting. There's a few other animals that have this negligible senescence concept, but it's pretty cool. Okay, so let's get started. There are two main parts of the cell cycle. First is called interphase, and the next is called M phase. We're going to focus mostly on interphase today, and then M phase is really when the cell divides. That's either going to be mitosis or meiosis. We'll spend the majority of the chapter looking at M phase, but today we're going to focus mostly on interphase because it's so short and easy to understand. 90% of the cell's life is spent in interphase. That's when the cell is doing whatever it is that it needs to do to survive, producing enzymes to digest food, if it's an intestinal or stomach cell, uh, helping carry oxygen if it's a red blood cell, producing pigment if it's a hair cell or a skin cell or eye cell. So 90% of the time, the cell is just doing what it's supposed to be doing and making protein. It can be separated into 3 distinct subphases: G1, S phase, and G2. So this pie chart down here is supposed to very, very simplistically show that this big strip on the outside, that's supposed to be a summary of interphase, and then these 3 larger subsections, that's going to be G1, S phase, and G2, and then this yellow part right here, that's going to be M phase. We'll spend most of our time looking at this. let's go to G1. G1, during G1, the cell is going to use mRNA to create proteins and organelles. This process is called gene expression and also known as protein synthesis. We learned about this concept in the DNA chapter. During G1, the cell grows in size, and DNA exists as chromatin or lower structures, as you see over here. This is because you want to have some areas of the DNA sequence exposed so that enzymes can read them and produce the proteins that they're supposed to produce, while other parts of the gene sequence will be wrapped up nice and tight and create this chromatin structure. During S phase, DNA is going to be replicated. S stands for synthesis, meaning it's going to make new DNA or copy new DNA molecules. Very briefly, during this process, DNA is vulnerable because base pairs will be slightly exposed while there's something called an Okazaki fragment that is unbound. And so mutations can occur here. It's very unlikely, but it can happen at this point in the cell's life cycle. Lastly, during G2 phase, the cell prepares for division. The number of organelles is doubled so that when the cell does divide, there'll be enough organelles to kind of jumpstart the process of the new life in the daughter cells. Enzymes double check the DNA and organize it into chromatids. So very complete, totally structured versions of DNA where there's no exposure. So the enzymes, it's basically turning off the cell process, this function of the cell, ever so briefly just so that it can divide. Chromatids are very closely related to chromosomes, but not exactly the same. So let's pause here and just kind of go over the differences between chromatids and chromosomes. I like to do that with an analogy. If an author writes a story and many copies of the books are sold, how many stories were made, it's kind of an interesting way to look at the difference between a story and a book. Let's say the story refers to the informational aspect, and the book refers to the physical aspect, the actual thing that you're holding, and the story refers to the thing that you're thinking about or analyzing. It's much more non-tangible. You can't really hold a story. You can hold a book, but you can't hold a story. So, in a similar method, that's how chromosomes and chromatids can be described. Chromatids are the physical coiling, the physical structure of a complete sequence of DNA tightly coiled and wrapped up. And the chromosome really refers to the genetic information or the story, the DNA story that's being told by a chromatid. So if you have a chromatid and it's totally, completely identical to another one, we call those sister chromatids. When they're together, they're linked by this thing called a centromere, and that creates our familiar structure of a chromosome. But if you wanted to, you could make the case that this is a chromosome as well because it has the same genetic story. It's got the genetic information, the DNA story, in this chromatid. So in that case, we're going to say our chromatid is the book, sister chromatids are copies, and our chromosome is the story. Okay, our last phase is M phase, and we're just going to briefly mention what happens here and go into much more detail in later lectures. During mitosis, the division of a single nucleus splits into two genetically identical daughter nuclei. You see that happening right here. Again, it might be lagging, so it's hard to see, but the red is one nucleus right here, and then it coils up and it goes through this process and it splits into two nucleuses at the end of this little animation 1, 2. After the nucleus splits, the cell itself will split, and that is known as cytokinesis. Cyto refers to the cytoplasm, the actual kind of liquidy gel that surrounds the nucleus, and then kinesis means to split. So first your nucleus will split, and then your cell will split. If you type in this YouTube URL, you'll get an interesting overview of what we just talked about. It's not the greatest detail.
Interphase.
It looks like this. It's by Mesquite Science, and the name is Interphase, and it basically just goes over everything we just talked about. Okay, thank you very much.